Chapter 7, Sexual Reproduction, Part 3. Why do you look similar to your parents, but not identical? It's because of sexual reproduction. Whereas asexual reproduction produces genetically identical clones, sexual reproduction produces genetically diverse individuals. As you already know, with sexual reproduction, both parents contribute half of the new organism's genetic material, so the offspring will have traits of both parents, but will not be exactly like either parent. Meiosis is another kind of cell reproduction. Organisms that reproduce sexually by joining gametes, which are ova or spermatozoa, in a process known as fertilization, must have a mechanism to produce haploid gametes. Haploid is a word that describes a cell that has half of the normal or general number of chromosomes. We have two main types of cells in our body. Another way to classify things, we have somatic cells, which are the body cells in general, like a liver or a heart or an eye. And then we have other cells that are considered to be germ cells, which are either those in the egg, in the ovaries and testes that will give rise to eggs or sperm, or the eggs or sperm themselves. Eggs and sperm, ova and spermatozoa, are haploid. They're haploid because two halves need to join together into a full new cell of a new life. So in this mechanism called meiosis, we have a type of cell division that halves the number of chromosomes in a cell. That happens in these germ cells in the ovaries or testes. During meiosis, then, the pairs of chromosomes separate and segregate randomly to produce gametes with one chromosome from each pair, instead of two chromosomes from each pair, as we would see in your eye cell or your heart cell or your liver cell. Meiosis involves two nuclear and cell divisions without interphase in between. And they start with one diploid cell and end up generating 4 haploid cells. Each division, named meiosis 1 and meiosis 2, has 4 stages: prophase, metaphase, anaphase, and telophase. These stages are similar to those of mitosis, but there are distinct and important differences. Introduction to meiosis. Prior to meiosis, in cells of the ovaries or testes, special stem cells called germ cells replicate their DNA, generating chromosomes with 2 sister chromatids. So a human cell prior to meiosis will have 46 chromosomes, 22 pairs of homologous autosomes, and 1 pair of sex chromosomes. Homologous chromosomes, or homologs, are similar in size, shape, and genetic content, and they contain the same genes as each other, although they may have different alleles of those genes. Alleles are versions of genes. So we might have a gene that says, be able to digest lactose, and we might have an allele that says, do not be able to digest lactose. They're both considered to be a gene for lactose metabolism. One allele says, yes, metabolize. The other allele says, no, don't metabolize. We'll see some more of this when we talk about inheritance. We have a figure here that shows another important concept called crossing over that we'll talk about in a little bit. But here we can see that we have the 2 chromosomes here in a pair. These would be considered homologous chromosomes. And they are, you can see they have letters on them. They have A's and B's on them. But as a pair, they're homologous. They have the same genes, although different versions. So the pink chromosome here has version capital A of its genes, the genes there that are duplicated, and the green or whatever turquoise chromosome there has the allele little a, and this chromosome also is representing what would be in a duplicated chromosome stage here. These are duplicated chromosomes, the X's that we saw earlier. And this is demonstrating some crossing over that we'll look at again. So we have these cells in humans then. We have 22 pairs of homologous chromosomes. 2 number 1s, 2 number 2s, 2 number 3s, etc. During meiosis, homologous or paired chromosomes separate and haploid cells form that have only one chromosome from each pair. So here we have we know that we've started in interphase to get to where we are in a cell in the ovary or testes, but we need to take these chromosomes and duplicate them. So we have them duplicated. So when we start in meiosis 1, then we're going to take that diploid cell. You can see that cell has 2 copies of each chromosome, a big red and a big blue, a little red and a little blue. You can see those little pieces of color on each one that are swapped over is representing crossing over. And then what we end up with then is that diploid cell dividing into 2 haploid cells. You can see the haploid cells have 1 big chromosome and 1 little chromosome now. But we're not finished yet, and we'll see how this works. This stage here still has to continue so that we can have more of these gametes. So we're going to look at, of course, having 2 haploid cells join into a diploid cell in the zygote. Now, so in this case here, we're actually popping apart each side of this chromosome and this chromosome like we saw before in mitosis, like reeling a fish in with a line. Of course, the fish is going to have to pop in too to do that. And we end up with then these separated chromosomes that are still full chromosomes, only they're half of the chromosome. That's okay though, because here the information was duplicated, so we can divide it in half and still have the same information. Here's a meiosis overview. So 2 cell divisions occur during the overall process. Let me just move this over a little bit if I can. Oh, won't let me do it. There we go. So a total of 4 haploid cells are produced. The 2 cell divisions are called meiosis 1 and meiosis 2. Meiosis 1 begins after DNA replicates, as you saw in the previous slide. Meiosis 2 follows meiosis 1 without DNA replicating again. Meiosis 1. We start with prophase 1, where the nuclear envelope begins to break down and the chromosomes condense. This is sounding familiar from mitosis in the cell cycle. Centrioles start moving to opposite poles of the cell and a spindle begins to form. Homologs pair up, which is a unique step in prophase 1. We didn't have homologs pairing up in mitosis. In prophase of mitosis and in meiosis 2, we don't have homologous chromosomes forming pairs. So here's what happens in a special step in prophase 1. It's called crossing over. So crossing over, also known as recombination, occurs during prophase 1, and it is the exchange of genetic material between non-sister chromatids of homologous chromosomes. So a chromosome that's been duplicated has 2 sister chromatids. And I refer to these sometimes as the arms and legs of a chromosome. So we can have in a duplicated chromosome, we have 2 arms and 2 legs. But if you take one of those arms and one of those legs on one side, that would be a sister chromatid. So this exchange of information is not among the chromatids on the same chromosome, it's between the chromatids of homologous chromosomes. This is the first event introducing genetic variation into the process of meiosis. During prophase 1, chromosomes condense and become visible, as we know, As the nuclear envelope begins to break down, homologous chromosomes move closer together. And this is now going to be interacted with something called the synaptonemal complex. Now, I'm not going to expect you to remember that word, but I want you to know, again, look at all the proteins that we've been talking about here. It's a lattice of proteins between homologous chromosomes, and it spreads to cover the entire length of the chromosomes. So this complex allows the tight pairing of homologous chromosomes in what's called synapsis. So everything is tightly associated, and we have the genes on the chromatids are tightly aligned with each other. The synaptonemal complex supports the exchange of chromosomal segments, which is an actual breaking off and reattaching between non-sister homologous chromatids in this process called crossing over. This exchange of genetic material can happen many times within the same pair of homologous chromosomes, creating unique combinations of genes. Following crossover, the synaptonemal complex breaks down and the cohesion connection between the homologous pairs is removed. So again, we have these proteins forming and then dissolving and then being used somewhere else for another reason. We have a lot of these proteins that are part of the cytoskeleton complex in the cytoplasm. At the end of prophase 1, the pairs are held together and are called tetrads because the 4 sister chromosomes of each pair of chromosomes are now visible. And here's another representation of crossing over. We just have a blue and a red chromosome. These are homologous chromosomes. We're looking at them. They're the same size, the same shape, but they're blue and red because these chromosomes have originally come from different individuals. So if you were to look at one of your cells in your cheek, for example, and we could color them according to what came from your father and what came from your mother, you would see in your cheek cells a blue and a red, for example, designating that. So every cell in your body is a combination from the very first time that the zygote was created, which resulted from the fusion of an ovum and a spermatozoa. And so we have these in all of our cells this way. When these chromosomes go through the crossing over process, then you can see on the far right that the homologous chromosomes are not fully intact as they were originally. They've swapped some information. In humans, there are over 8 million configurations in which the chromosomes can line up during metaphase 1 of meiosis. It is the specific process of meiosis resulting in 4 unique haploid cells that results in these many combinations. Now there's a mathematical calculation for this. It has to do with the number of chromosomes, actually the number of pairs of chromosomes, so 23 pairs, and then the different ways that they could actually arrange themselves in the cell as it's dividing. So we had crossing over as the first exchange that puts variety in sexual reproduction. And now we have independent assortment, which is another way to put variation into sexual reproduction. Okay, so in this process here, the chromosomes inherited from either the father or mother can sort into any gamete, and it produces the potential for tremendous variation. So during metaphase 1, the tetrads move to the metaphase plate. Now, you remember the metaphase plate would be, I think we referred to it this way before, the equator of the cell with the kinetochores, which are points on the chromosomes themselves, facing opposite poles. And so these homologous pairs are oriented here at the equator randomly. Nobody knows how to predict how they're going to line up. So it could be that we have a red and a blue next to each other, and then under that we have a blue and a red next to each other, and then we have a red and a blue next to each other. And these are going to be pulled apart to opposite poles. So we might, at one pole, we might have all blue, for example, of course, just oversimplifying. And on the other pole, we might have all red. But we might have 22 blues and 1 red. or we might have 11 blues and 12 reds, or something like that. So there's many, many combinations of how that could work. And I can show you an example here. So this is in metaphase 1. The spindle fibers attach to the paired homologous chromosomes, and the paired chromosomes line up along the equator, or they call it the metaphase plate. This only occurs in metaphase 1. In metaphase of mitosis and of meiosis 2, it is only sister chromatids that line up. And we'll see that in a minute, so just hang on. You can see here in these two scenarios, the top and the bottom. The top one, we have a blue next to a red, and then we have a blue next to a red in the center cell there. The bottom one, we have a blue next to a red and a red next to the blue. And as these chromosome pairs are pulled apart, it gives a different result in the next cell that comes from that. So that top cell divides into two, and then depending on where the reds are and where the blues are, it makes a difference in the outcome eventually to what the actual gamete, the ovum or the spermatozoon, is. And of course, those then will be recombined with another gamete. Independent assortment then is the second mechanism that introduces variation into the gametes or spores. This happens in other types of species too. In each cell that undergoes meiosis, the arrangement of the tetrads is different. The number of variations is dependent on the number of chromosomes making up a set. So with our number of sets, we have a certain number of combinations that are possible. If an organism had 10 pairs, that would be a different possibility of the number of arrangements that could be made. So there are two possibilities for orientation of each homologous pair at the metaphase plate. We can call them left and right. And we can, if you look back at this one here, we can see how we have a left and a right option on each of those. Okay, continuing through metaphase 1, meiosis 1. In anaphase 1, the spindle fibers shorten and the chromosomes of each homologous pair start to separate from each other, similar to what we see in mitosis. One chromosome of each pair moves toward one pole of the cell, and the other chromosome moves toward the opposite pole. But in this one, in anaphase 1, you might notice that we're not separating the chromosomes into their separate arms and legs, so to speak. The whole chromosome is moving. Then we have telophase and cytokinesis. We have the spindle breaking down and new nuclear membranes forming. The cytoplasm of the cell divides and two haploid daughters result. The daughter cells each have a random assortment of chromosomes with one from each homologous pair. And now both of these will go on to meiosis 2. So they are haploid already, but in the efficiency of nature, why don't we take that information, which is actually duplicate information, because these chromosomes after meiosis 1 have 2 arms and 2 legs. Why don't we split them in half and get double the genetic material to go on to reproduce? So in meiosis 2 then, the nuclear envelope breaks down again, the spindle begins to form in each haploid daughter cell. The centrioles also start to separate. Now in metaphase 2, the spindle fibers line up the sister chromatids of each chromosome along the equator. And then in anaphase 2, the sister chromosomes separate. This is, again, we're reeling the fish in to opposite ends of the cell, popping the fish apart and reeling it in. Then telophase and cytokinesis. The spindle breaks down and new nuclear membranes form. The cytoplasm of each cell divides and 4 haploid cells result. Each cell has a unique combination of chromosomes. So here's an illustration of how that works. We saw this earlier in this set of slides here. The interphase in the cell will go through the DNA synthesis so that by the time we get to meiosis 1, we have a cell that is has duplicated, replicated, in other words, chromosomes. That represents replication of DNA. And then through meiosis 1, this cell, which has 2 pairs of homologous chromosomes, will now have one of each homologous chromosome in the daughter cells. So instead of having two larges and 2 smalls, we have 1 large and 1 small in each one. And then going through meiosis 2, those duplicated chromosomes now pop apart at the centromere, and their genetic material is spread out to result in 4 haploid cells.